Hi everyone, my name is Kaylani and I'm a science editor here at Smart Edition Academy. In this video, we're going to go over 10 science questions that cover material like you would see on your test. But before we get started, make sure you subscribe to our channel and ring the bell for notifications so you get notified when more videos like this come out. Also, make sure to check out the links in the description below to some of our great free resources. There, you're going to find Smart Edition's free practice test, our Facebook study guide group, which has tons of other great resources to help you study. And if you want more practice, you can check out the link to Smart Edition online course, where you actually have access to practice tests, lesson modules, video lessons, and other flashcards. Some really great stuff that's going to help you study. But otherwise, let's go ahead and get started. Which sequence describes the hierarchy level of biological organization? A. Kingdom, phylum, class, order, family, genus, and species. B. Genus, class, kingdom, species, order, phylum, and family. C. Family, species, genus, order, kingdom, class, and phylum. Or D. Species, kingdom, genus, class, family, phylum, and order. So take a moment to think about this. But if you'll recall from our material, this taxonomic system is just something you kind of have to memorize. So actually the answer is A. And that's because the taxonomy is the process of classifying, describing, and naming organisms, and that there are 7 levels in the Linnaean taxonomic system, starting with kingdom and going all the way down to species. The next question, which example is part of the scientific method? A student reads about a new way to harness energy from the sun. B, a researcher studies the effects of car exhaust on how people breathe. C, a researcher analyzes how many plants respond well to a new fertilizer. Or D, a student discovers how insulin plays a role in the development of diabetes. Once again, take a moment to look at your answer options, but try to think back to what the scientific method really is. You have to identify a problem, you have to conduct some research, formulate a hypothesis, and then conduct an experiment, observe that experiment over, and come to a conclusion. So which answer option best communicates that? Answer option C, a researcher analyzes how many plants respond well to a new fertilizer. And why is that? Well, that's because one of the steps that I identified is that you have to analyze information or data collected from the experiment to conclude whether the hypothesis is supported.
Welcome to Smart Edition's video tutorial on cell structure, function, and type. In this video, we will cover material that will better explain the cell theory and compare different types of cells. Let's get started. A cell is often referred to as the smallest unit of life because at even this basic level, all functions of life can be performed, from responding to stimuli to obtaining and using nutrients. The invention of microscopes and improvement in lens production provided the technical advance for scientists like Robert Hooke to observe what had once been invisible, with early 17th century microscopes similar to the one built by von Leeuwenhoek. In fact, the term cell was coined by Robert Hooke in 1665 because as he observed cork under a microscope, he made the observation that the structures he was observing looked like small compartments or rooms of a monastery. The idea that cells are the basic unit of structure and function developed from the fundamental tenets of the cell theory. Most notably, the cell theory was established by the work of several scientists, including Theodor Schwann, Matthias Schleiden, and Rudolf Virchow in the 1830s, who concluded that 1. All living organisms are composed of one or more cells. 2. The cell is the basic unit of structure and organization in organisms. And 3. Cells arise from pre-existing cells. Regardless of whether a cell is classified as prokaryote or eukaryote, the cell theory applies. Classification of cells as prokaryotes or eukaryotes depends on the presence or absence of specialized subcellular structures called organelles that have evolved to carry out life functions, including protein synthesis and cell respiration. Prokaryotes are simple, unicellular organisms that lack membrane-bound organelles, are classified as members of the domain Bacteria and the domain Archaea, and are significantly smaller than eukaryotes, with a typical size range less than 5 micrometers. There are approximately 9,300 species of bacteria, and microbiologists broadly classify them through a common Gram staining technique used to differentiate bacteria based on the composition of their cell wall. While prokaryotes lack membrane-bound organelles, they do share some organelle similarities with eukaryotes, including a plasma membrane and ribosomes. In some cases, prokaryotes and eukaryotes also have flagella and a cell wall in common, albeit composed of different chemical constituents. One structure that eukaryotes do not possess is the presence of pili. These structures in bacteria are necessary for a version of sexual reproduction called conjugation, that facilitates the transfer of DNA between bacterial cells to increase genetic variation in the population and can confer resistance to multiple antibiotics. Conversely, eukaryotic cells comprising the domain Eukarya and the kingdoms Fungi, Plantae, Animalia, and Protista can range in size from 10 to 100 micrometers and vary structurally from their prokaryote counterparts. While prokaryotes do not contain membrane-bound organelles and lack a nucleus to house DNA, eukaryotes contain an assortment of organelles, though their number and kind will vary. In addition to the presence of a nucleus, eukaryotes also contain ribosomes, just like prokaryotes, but these ribosomes can be classified as either attached or free. While both forms participate in protein synthesis, free ribosomes are involved in products that will be used by the cell directly, while ribosomes attached to an endoplasmic reticulum participate in the synthesis of protein products that will be exported by the cell. In addition to ribosomes, organelles that are uniquely eukaryotic include the Golgi apparatus, endoplasmic reticulum, mitochondria, lysosomes, vacuoles, and nucleolus. The endoplasmic reticulum comes in two varieties, smooth and rough. Smooth ER is involved in the synthesis of lipids like steroid hormones, while the rough ER is directly involved in protein synthesis and is studded with ribosomes. Products from the ER are subsequently sorted and packaged by the Golgi apparatus, whose function is often compared to as a package delivery service. The nucleolus, found in the nucleus, is responsible for the production of ribosomes, while lysosomes store hydrolytic enzymes responsible for intracellular digestion and are often referred to as the garbage disposal of the cell. In fact, during the process of endocytosis, when a cell engulfs materials from the environment, it becomes packaged in the form of a vacuole and then subsequently merges with a lysosome to facilitate digestion. Lastly, mitochondria, often called the powerhouses of a cell, carry out the process of cellular respiration, a set of metabolic reactions that convert biochemical energy in the form of nutrients like sugar into ATP. This concludes Smart Addition's video tutorial on cell structure, function, and type. Please refer to the accompanying text for additional content support.
Next question. Why did it take many years for the cell theory to be developed? A. Advancements in microscopy took place slowly. B. Cells were difficult to isolate after experimental analysis. C. Researchers believed a cell formed from pre-existing cells. Or D. Scientists already proved that cells were essential for life. So take a moment to look at your options again, but let's recall what the cell theory is. There's three parts to it, right? All living things are composed of one or more cells. Cells are alive and represent the basic unit of life. And all cells are produced from pre-existing cells. Also option C. So likely our answer isn't going to be one of the components of that cell theory. So remembering that Robert Hooke actually discovered the first cells in the mid-18th century, our answer option for this one is going to be A. Cell theory is a theory because it is supported by a significant number of experimental findings, but it took many years to be developed because microscopes were actually not powerful enough to make such observations. All right, question 4. What organelle is only associated with plant cells? A. Cell wall. B. Ribosome. C. Cytoplasm. D. Golgi apparatus. So looking at our answer options, it's pretty clear if you memorized your cell organelles that our best answer option is actually going to be A. Cell wall. And that's because only plant cells have cell walls, which help protect the cell and provide structural support. What phase of the cell cycle must happen before cell division? A. Interphase. B. Metaphase. C. Prophase. Or D. Telophase. So let's recall that the cell cycle is actually a circular process which regulates cell division, and that it can be divided into two major phases, interphase and the mitotic or M phase, which actually includes options B, C, and D, metaphase, prophase, and telophase. So what does that leave us? With answer option A. And that's because before mitosis or meiosis can occur, interphase must happen. Next question. What is the correct order of the stages of the cell cycle? A. G1, S, G2, M. B. G2, S, G1, M. C. M, S, G2, G1. Or D. S, M, G1, G2. So let's do some basic elimination processes here. So we obviously can kind of infer 1 comes before 2. So let's just eliminate option B and C at the moment because G2 coming before G1 just doesn't seem like a likely option. So we're left with G1, S, G2, or M, or SM, G1, G2. Now, this is just one of those things you have to kind of memorize the cell cycle. So if you recall our chart, you're going to remember that the G1 is actually the growth phase, and that's the first gap phase, which prepares the cell to copy its DNA. And it actually goes into the S phase, which includes DNA replication, before it goes into the G2 phase, which is the preparation for division, before hitting the M or mitotic phase that we previously talked about, which includes all of those stages from prophase all the way to cytokinesis. So our answer option is A. And that's because during interphase, the cell undergoes an initial gap phase called G1 before its DNA is copied in the S phase. And after that, it's copied and goes into the second gap phase, like we already said. And then the cell is ready to enter into the mitotic phase, which divides into daughter cells.
Welcome to Smart Addition's video tutorial on cellular reproduction. In this video, we will be providing an overview of different forms of reproduction, as well as discussing the similarities and differences between mitosis and meiosis. Let's get started. One of the most important characteristics of life is reproduction, the process of making more of something. In the case of living things, reproduction can occur in several ways, depending on the species. For example, sexual reproduction, the most common reproductive strategy among multicellular organisms, is defined as the fusion of special reproductive cells called gametes that contribute equal amounts of genetic material donated by male and female members of a species. The result of this fusion is the formation of offspring with a complete set of genetic material or chromosomes. This reproductive strategy is advantageous because mixing genetic information leads to variation among members of a species, and this natural variation, as described by the theory of evolution, is the raw material for natural selection. However, not all species reproduce sexually. Asexual reproduction does not involve the fusion of gametes. Instead, asexual reproduction produces offspring from a single parent cell. There are a number of types of asexual reproduction, including binary fission and budding. While both produce offspring from a single parent, binary fission is a common reproductive mechanism among bacteria and single-celled protists, while budding is most often associated with yeast and the animal hydra. Both forms of asexual reproduction produce offspring with a complete set of genetic information. Binary fission leads to the formation of two identical daughter cells, while budding produces a single, small daughter cell that becomes independent of the parent cell by pinching off. Interestingly, while asexual reproduction is the preferred mode of producing offspring among bacteria and yeast, they're both capable of carrying out a variation of sexual reproduction called conjugation, whereby genetic material is exchanged between cells, usually under unfavorable environmental conditions. As mentioned, the multicellular animal hydra propagates by budding, while many multicellular organisms, including humans, rely on sexual reproduction to produce offspring by combining genetic contributions from mom and dad. They also rely on asexual reproduction to repair and replace old or damaged somatic or body cells. They accomplish this through mitosis. It's worth noting that mitosis is only one stage in the broader cell cycle consisting of interphase, when a cell grows and replicates its DNA, and M phase, or mitotic phase, when a cell divides its DNA in cytoplasm into two new identical daughter cells. A typical somatic or body cell is not actively dividing. Cells that are not actively dividing are said to be resting in the G0 phase. Of course, the term resting doesn't mean the cell is inactive or asleep. It refers to the normal activities and functions that the cell is programmed to carry out. The image depicted to the left shows the complete cell cycle. As mentioned, cells do not enter the cell cycle unless prompted to do so. If a cell does receive a signal to initiate the cell cycle, it will first enter interphase, which consists of three substages, G1, S, and G2. During the period of time referred to as G1, the first gap period, the cell grows in size and assembles the cellular material and machinery it will need for subsequent stages. Following the G1 phase is S phase, or the synthesis phase. During this stage, DNA replication takes place to ensure that the cell has enough DNA to distribute to two new daughter cells. Please refer to Smart Addition's video lesson on genetics and DNA, Part 2, for more details regarding the process of DNA replication. After successfully replicating DNA, the cell enters the final stage of interphase called G2, or the second gap period, where the cell organizes cellular contents and generates additional cellular machinery in preparation for M phase. At the conclusion of interphase, the cell is now ready to begin evenly dividing its replicated DNA and its cytoplasm for the production of two new daughter cells. M phase of the mitotic phase begins with mitosis, the process of nuclear division, and concludes with cytokinesis, the equal division of the cytoplasm. To ensure that diploid number is maintained and the chromosome number conserved, the cell will participate in a series of 4 stages that promote the even distribution of replicated DNA. Recall that the end product of DNA replication, which takes place during S phase, is 2 complete sets of DNA. During prophase, the first stage of mitosis, the replicated DNA condenses into visible structures called chromosomes. Their microscopic appearance resembles an X and are more specifically referred to as a replicated chromosome because each arm of the X corresponds to a genetic copy of the original parent cell DNA and are referred to as sister chromatids. In other words, one replicated chromosome is composed of 2 sister chromatids. In addition to replicated chromosomes becoming visible during prophase, the nuclear envelope of the dividing cell begins to degrade, and the replicated centrioles, which are only found present in animal cells, begin to emerge and initiate their migration to opposite poles of the cell. The centrioles play an integral role in the attachment and eventual migration of chromosomes during later phases, as well as in the elongation of the cell in preparation for cytoplasmic division. The second stage of mitosis is metaphase, where replicated chromosomes align themselves vertically along the established spindle apparatus formed by the centrioles. At this point during mitosis, the centrioles have completed their migration to opposite poles of the cell and have successfully formed a spindle apparatus similar to a clothesline for the attachment of replicated chromosomes. During this stage, the central part of each replicated chromosome, called the centromere, surrounded by a kinetochore complex and bound together by the protein cohesin, connects with the kinetochore microtubule, while non-kinetochore microtubules elongate the cell. During this stage, it is important that the chromosomes are properly attached and aligned at the metaphase plate, because once chromosome migration is initiated, it can't be stopped. If one side of a replicated chromosome does not attach to a kinetochore microtubule produced by these centrioles, the cell runs the risk of abnormal separation, which may result in the uneven distribution of chromosomes, where one daughter cell has one additional chromosome and the other has one fewer. Once the replicated chromosomes have been checked for alignment and attachment, they enter anaphase, the shortest of the mitotic phases. During this phase, the replicated chromosomes begin their separation and migrate toward opposite poles of the cell. Migration is facilitated by the activation of the enzyme separase, which digests cohesin that binds these sister chromatids together. Once cohesin has been degraded, the now single unpaired chromosomes begin their migration. Motor proteins at the kinetochore attach and chew back the microtubule, essentially pulling the single unpaired chromosome along as the microtubule depolymerizes. In this way, each chromosome is pulled toward opposite poles of the cell, as opposed to being kind of like reeled in at the site of the centrioles. As chromosomes migrate, the cell membrane begins to pinch at the center of the cell, initiating cytokinesis. The last stage of mitosis is telophase. During this stage, genetic material has successfully made its way to opposite poles and becomes less condensed. The nuclear envelope begins to reemerge, thus concluding mitosis. Of course, the even distribution of cytoplasm must also occur in order that 2 identical daughter cells are formed, and is accomplished by the formation of a cleavage furrow produced by a contractile ring made of actin filaments. Essentially, this contractile ring squeezes the cell membrane inward, eventually separating the 2 daughter cells. At the conclusion of mitosis, the 2 new cells may return to normal cellular function in G0 or may continue dividing, depending on their broader cellular function. In either case, genetic continuity is guaranteed. It's important to note that while this process occurs in plant cells, it does vary slightly. Specifically, plant cells do not use centrioles to build and organize their spindle apparatus, and they also do not separate their cytoplasm by the formation of a cleavage furrow. Instead, they produce a cell plate that emerges between daughter cells, which eventually aggregates to form a mature cell wall. It is important to note that passage through the stages of the cell cycle are heavily regulated by cyclins and a family of enzymes called cyclin-dependent kinases. This high level of cell control ensures that the cell is large enough to divide, has accurately copied DNA, and has aligned and attached replicated chromosomes to guarantee even migration to opposite poles. Mitosis is not the only process that evenly divides genetic content of a nucleus. While mitosis guarantees genetic continuity, the process of meiosis guarantees genetic variability. Meiosis takes place in the cells of reproductive organs and results in the formation of 4 daughter cells, called gametes, each containing a haploid or half the number of chromosomes, and proceeds through 2 separate divisions. This makes sense when we consider the purpose of meiosis in the context of sexual reproduction. In other words, when reproductive or haploid cells from mom and dad fuse during a reproductive event, they generate an offspring with a complete set of chromosomes known as the diploid number. This number varies across species, but for humans, a diploid number of 46 is achieved from the contribution of 23 chromosomes from mom and 23 chromosomes from dad. Often referred to as gametogenesis, meiosis is preceded by interphase, just like mitosis. In fact, the stages of meiosis are also very similar with important differences. Most importantly, the cells undergo 2 divisions, namely meiosis 1 followed by meiosis 2, in order to achieve the haploid number characteristic of reproductive cells. Let's assume that these special reproductive cells have undergone interphase and are now entering the first division called meiosis 1. Meiosis 1 begins with prophase 1, where the nuclear envelope disappears and the replicated chromosomes become visible. So far, there are no glaring differences between prophase 1 of meiosis 1 and prophase in mitosis. However, during prophase 1, replicated chromosomes will undergo a process called synapsis. During synapsis, homologous chromosomes, these are replicated chromosomes that carry the same genes but are classified as maternal or paternal, will fuse to form a tetrad, so named because it consists of 4 chromatids, 2 sister chromatids per homolog. The resulting tetrad will often undergo a process called crossing over. Crossing over occurs when the arms of chromatids become so entangled that they break and reform, causing an exchange of genes between homologous chromosomes. Ultimately, crossing over and crossover events increase variation among offspring through the recombination or mixing of paternal and maternal genes. The second stage of meiosis 1 is metaphase 1. Just like mitosis, metaphase 1 is responsible for the alignment of chromosomes along the metaphase plate. The difference in metaphase 1 is that each tetrad formed in prophase 1 will align, not individual replicated chromosomes. Once alignment and kinetochore microtubule attachment have taken place, the tetrads are prepared to undergo anaphase 1. During anaphase 1, homologous pairs will split so that each homologous pair making up the tetrad migrates to opposite poles of the cell. Once the homologous chromosomes have reached their respective poles, the nuclear envelope begins to reform and the chromosomes become less visible during telophase 1. Cytokinesis follows telophase 1 by evenly dividing the cytoplasm. The 2 cells formed at the end of meiosis 1 have achieved the haploid number of chromosomes, but the chromosomes are still in their replicated chromosome state. This, of course, will require a second division, called meiosis 2. Meiosis 2 proceeds through the same stages as meiosis 1, namely prophase 2, when the replicated chromosomes become visible and the nuclear envelope disappears, metaphase 2, when replicated chromosomes and not tetrads align vertically along the metaphase plate, similar to mitosis, anaphase 2, when the replicated chromosomes split and single unpaired chromosomes migrate to opposite poles along the spindle apparatus, and telophase 2, when nuclear division is complete. Meiosis 2, just like meiosis 1, is followed immediately by cytokinesis when the cytoplasm of dividing cells is evenly separated. It is important to note that meiosis 2 is not preceded by interphase. If it was, the reproductive cells would never achieve their single unpaired chromosome state required for sexual reproduction. The end result of meiosis 2 is 4 cells that contain a haploid number of single unpaired chromosomes. The formation of reproductive cells is often referred to as gametogenesis because the end products are called gametes. Of course, gametes can be classified as sperm or egg cells depending on their origin. If gametogenesis results in the formation of sperm cells, it is specifically referred to as spermatogenesis, while the formation of egg cells is called oogenesis. Both processes result in single, unpaired haploid cells. Spermatogenesis produces 4 mature sperm cells, while oogenesis results in the formation of 1 mature egg cell and 3 polar bodies. These are small haploid cells that don't develop into mature egg cells. In summary, both mitosis and meiosis are preceded by interphase, where cells grow and DNA is replicated. Both begin with a single parent cell and produce new cells. The biggest differences involve the class of cells that undergo each process. Mitosis produces identical diploid somatic cells, while meiosis produces non-identical haploid reproductive cells. Both mitosis and meiosis undergo prophase, metaphase, anaphase, and telophase. Mitosis requires 1 division to achieve diploid cells, while meiosis requires 2 divisions to achieve haploid cells. Lastly, mitosis guarantees genetic continuity, while meiosis encourages genetic variation accomplished during crossing over. This concludes Smart Addition's video tutorial on cellular reproduction. Please refer to the accompanying text for additional content support.
All right, next question. When would a cell most likely contain the most nucleotides? A, S phase, B, G1 phase, C, M phase, or D, G2 phase? So let's recall that cell cycle process again. So we just identified that the G1 is that growth phase, the S is the DNA replication phase, or G2 is the preparation for division, and that M phase is actually the division for daughter cells. So when would it most likely contain the most nucleotides? Answer B. A cell copies its DNA during the S phase, and the nucleotides are the building blocks of the DNA. So the step preceding the S phase, which we know was the G1 phase, is actually the phase of the cell cycle when it would contain the most nucleotides. Next question. The sequence of amino acids in a gene determines A, the primary structure of a codon, B, the primary structure of a protein, C, the primary structure of a nucleotide, or D, the primary structure of a nucleic acid. So let's recall how DNA structures work. So the DNA is that double helix structure that wraps around and is actually formed from nucleotides. So definitely not C, the primary structure of a nucleotide, right? So let's eliminate that one. So then what does DNA code for? Codes for protein. Next question. Which statement best represents Mendel's experiments with garden peas? A, as a result, Mendel developed several theories that have since been disproved. B, Mendel realized he was on an incorrect track, which led him to other experimental media. C, as a result, Mendel developed foundational conclusions that are still valid and followed today. D, Mendel collaborated with others interested in genetics to develop hereditary guidelines we still use today. So looking at our options and remembering our material, Mendel is actually the father of genetics, and he actually had a theory of heredity. So where does that leave us? With option D. Mendel developed theories of genetics that scientists around the world still use. And that brings us to our last question. Mendel discovered the pattern associated with blank after developing a series of rules in genetics. A, epigenetics, B, heredity, C, heterogeneity, or D, taxonomy. And I think we already know the answer to this one, but it's going to be option B. Mendel was accurately able to predict the patterns of heredity by studying the rules related to genetics, and these rules helped shape his theory of heredity.